Alright, guys, welcome you all to this video. Let's talk about biochemistry practical. And firstly, let's talk about carbohydrates, right? Carbohydrates, what thing you must know and what things you must not focus on in order to prepare well for tomorrow's exam, right? See now, all the principles and procedures will be given in boards. Right now it will not be given, only to intimidate you, but that must not make you nervous. All right, for principles, what you must know is, for these very experiments, not all the experiments you must know. That is, there are many experiments which are enlisted in your log book, but they are not very important. All right, only these 7 or 8. Let's come down. Possible confusions. What can be the possible confusions in different tests? Firstly is Osajon test. Osajon test, always remember, @MS. Everyone plays and watches cricket. Everyone must know the name of cricketer called MS Dhoni, right? From with that I have picked out MS. Maltose is always sunflower shaped. Automatically the other one, lactose, becomes puffball or hedgehog crystals. Always remember, while writing on the paper, you must be careful about the spelling. Hi. Always be careful about the spelling. Now let's talk about Barfoed's test. Barfoed's test, there is a disaccharide and monosaccharide. Now people generally confuse on which coloration to be provided to which saccharide, because disaccharide has more number of saccharide molecules, that is sugar molecules, and we suppose that more number of sugar molecules, more will be the coloration, and so for disaccharide we give deep coloration, but that is not the case. The case is thaka opposite. For disaccharide we must give light blue coloration, right? For monosaccharide, there must be deep blue coloration. Now, what can be the possible questions in carbohydrates that can be asked in table VIVA? See, up to the things that I have enlisted up to now, like principles are the ones that are asked most frequently. Firstly, they will ask you principles, and then they will ask you what is your outcome, if your test is positive or negative. See, you must be careful that all your tests should be positive, all right? Or your test, whichever you are doing, like say for example, starch. Right, for starch, all the tests that you have enlisted in your logbook, the results you find out must be similar to that. One test you do not do according to what should be coming, then your marks will be deducted. All right, so please be careful on that. Firstly, principles can be asked in the table VIVA. Another thing that can be asked in table VIVA is obviously this question: why Benedict test is called semi-quantitative test? The things which are written in note in your logbook, remember, note, NOTE, always pay attention to them as well, right? Why Benedict test is called semi-quantitative? Why? Because it does not give the exact value of reagent required. Reagent ko exact value not diye pani. What it does is, it gives relative percentage of concentration of gradient. That is, for example, 0.5%. We have violet coloration. In the same way, if you see plus will give different type of colorations, ranging from what? Timro violet, blue, red, brick red. All right, so you must be careful on that. Let's talk about proteins. Proteins also, you must know all the principles of all the tests. I must not be repeating it. Yes, in different experiments there are particular questions you must be careful about. Like, first question can be asked from half saturation and full saturation test. Salting in, and what is the meaning of salting out? You know, salting out you all know about. That is adding salt concentration, and higher the salt concentration, it will remove the water from the protein surface. And if water is removed from the protein surface, protein-protein interaction increases, and thus precipitation of protein occurs, which is called as salting out. While as salting in means lower concentration of salt stabilizes and solubilizes protein. Bhaneko, in a beaker, if there is lower concentration of salt, राइट, तो यहाँ प्रोटीन होला, यहाँ प्रोटीन होला, यहाँ प्रोटीन होगा, है ना? तो उसके बीच में यदि थोड़ा थोड़ा प्रोटीन है, तो क्या करेगा कि ये प्रोटीन का स्ट्रक्चर को स्टेबलाइज कर देगा. इट विल स्टेबलाइज द प्रोटीन स्ट्रक्चर, जसको कारण चाहिँ के हुन्छ, इट विल मेन्टेन इट्स सल्युबिलिटी. त्यसलाई के भनिन्छ? सल्टिङ इन. सल्टिङ इन, इट इम्प्रुभ सल्युबिलिटी. सल्टिङ आउट, इट डिक्रिजेस सल्युबिलिटी. द्याट इज द मेजर डिफरेन्स. Heat coagulation test, Millions test, aldehyde test, Sakaguchi test, all the tests you must know principle and the points which are enlisted in note. That is very important. All right. Now the other question that can be asked, other kind of weird question that can be asked in protein identification can be lead acid test, where it is said that for sulfur containing amino acid, we all know about that, right? But there are two amino acids which give this test, that is cysteine and cysteine. What is the difference between cysteine and cysteine? Cysteine, EINE. Always remember, the one with the E is an amino acid, while as the one with no E is an amino acid joined by disulfide bonds. I'm sorry, it is disulfide bonds. All right. Disulfide bonds. Generally, what kind of bonds are present between amino acids? Dipeptide, but in case of cysteine, disulfide bonds are present. Come down. Remember, peptone and gelatin. If your sample is peptone or gelatin, never be confused. In peptone, all the color reactions are positive, while in gelatin, negative aldehyde and acetate test. Also remember one thing, gelatin is a type of derived protein. Now, why is it called a derived protein? Do you know of that? Because it is derived from denaturation, or we can call it denaturation of which protein molecule? Of collagen. I know my handwriting is atrocious, plus please be cooperative with it. Now see, if you guys know these things, you can almost do good in your examination of identification. Now let's talk about things for VIVA. Obviously you will be asked from both paper 1 and paper 2, but what things to remember in one night? You can't just read everything, so please pay attention. Carbohydrates, paper 1 basic carbohydrates. We must not forget that everything that you study, you must know the classification about it. All right, so carbohydrates, you must know the classification. Also definition of anomers, epimers, enantiomers and reciprocal mixture. I will not say the definition, you must read it via your notes or fast track. In anomers, you must remember that A for anomers, and since A is the first alphabet, so it is always change in configuration at C1 level, at the first carbon, and any change in configuration other than C1 will be called as epimers. For example, glucose and galactose. I don't exactly remember, but it must be C4, I think, or it can be C2 as well. Find it. All right. Amino acid, you must know about classification. Isoelectric PPT, obviously you will study in the principle, that is isoelectric PPT test, right? The pH at which the pH isoelectric PPT, rather it should be called as isoelectric pH, the pH at which concentration becomes high and solubility becomes least, due to which precipitation is most evident. All right, now let's come down. You must know about the properties of peptide bond. All right, bipeptide bond, and what are the properties of it? It should be rigid, polar, trans configuration must be showing, and partial double bond character must be present out there, right? In proteins, you must know about classification, and classification in great detail. All right, you have functional classification, you have structural classification, right? In functional classification, you must remember the examples of each. For example, what are the example of transport proteins? We call it as ovalbumin, right? We call it as what? Ovalbumin. So you must know such kind of functions, so that if you are asked about functions of proteins, you can answer them as well. Now let's talk about other question that was asked in our time, and it might be asked in your time as well. Function of albumin or histidine. You can say about albumin, obviously you can do, or obviously you do, right? But in case of histidine, always remember it is important in buffer 1. Other, it is important constituent of hemoglobin. You must point out these two differences. All right, biologically active forms of amino acids and proteins also you must care about, right? Now let's talk about lipids. You must know classification, functions of phospholipids. It was asked very frequently, it might be asked in you as well, so please pay attention to this. Vitamins, you must not know anything. Like vitamins intimidated me the most, right? It might confuse you as well, so don't read anything at all. Just pay attention on one basic thing, that is classification. All right, enzymes. In enzymes, you must know classification and other things as well. Factors affecting its activity, and inhibition is the must. Inhibition you must know about. Competitive and competitive, you must have studied this in pharmacology as well. You can just correlate and study. Other important thing is isoenzymes and examples. Where are isoenzymes located? For example, creatine kinase, CK. You will be asked examples about it. CKMB, CKMM, CKBB, right? CKMB in heart, CKBB in brain. Like as examples, you can also give about lactase dehydrogenase. You can give examples about aldolase, right? You can give many examples. Or also allostatic inhibition. What question I was asked personally most frequently was, what do you mean by allostatic site? Any site on the enzyme other than main site where the substrate is acting is called as allostatic site, right? And what do you mean by allostatic inhibition? You must know about that as well. Paper 1 genetics. There are many things in genetics. Don't study all the things, rather remember what do you mean by replication, transformation or transduction. All right, process is hardly asked. Role of enzymes are asked rather. What is the role of topoisomerase in replication? What is the role of helicase in replication? These kind of questions are frequently asked. Post-transcriptional, post-translational changes you must know about. Post-transcriptional is the most frequent. We all know about it. There is capping, right? There is slicing, and obviously there is poly A tail. You must know about these. Only the definitions might work. You must not know in detail. All right. Replication. What is the difference in prokaryotes, eukaryotes? All right. For example, what prokaryotes we have single oocyte. In eukaryotes we have multiple oocyte. We have DNA polymerase 1, 2, 3. Right. In eukaryotes we have what? DNA polymerase alpha, beta, delta, gamma, and eta. There are different enzymes working for different roles. Restriction endonucleases and examples. Restriction endonucleases, you must know the definition, and in examples you can give many examples like Taq polymerase, BAMH1, EcoRI. You can give many examples. All right. The most important, which I would suggest you to study, is tumor markers, because tumor markers are the most frequently asked questions. Tumor markers, you must know about the features, limitations, and examples. Examples I must not tell you, or I need not tell you, because there are many that you already know of. CA125, ovarian, PSA, prostate. All right. Such examples must not be forgotten. All right. In case of features and limitations, you must know via your notes. These are not given in fast track. I must say that as well. In features, obviously cheap, cost effective, high sensitivity, high specificity, constant in non-disease, and remains low in remission phase. All right, these are 4 or 5 important features which I have listed down for you. You can rather screenshot it and study. All right. In paper 2, what can be asked? Paper 2, you must not focus too much, because paper 2 is not very asked. But if they are asked, they will ask about what are the uses of neurotransmitters like acetylcholine, catecholamines, polyamines, right? Not apart from this. Serotonin, or you may say other like GABA or dopamine, these are hardly asked. All right. You must know about general neurotransmitters and why they are called neurotransmitters. Integumentary, melanin. The process of melanin synthesis you must know, and complications by it. For example, albinism. All right. Why does it happen? What happens in that? You must know that. And in case of MSK, there is molecular mechanism of skeletal muscle contraction, an important thing. Other can be biomarkers. Biomarkers are the same thing, like myoglobin, all right, like CK, like lactate dehydrogenase, like aspartate transaminase. All right, these are some biomarkers which you must study. What are the isoenzymes? Where are they present? How fast they act? This kind of things might help. Quiz, role of calcium in homeostasis. It is not asked rather, but you can be asked like details about it. Like when is calcium increased, in which conditions, conditions calcium is decreased. All right, so these will help you out, I hope. Let's stop the video right here.